what is the regulation of glycolysis? How is glycolysis regulated? Don't forget, this one is very, very important. So how is glycolysis regulated? First of all, we're talking about the dietary regulation, we're talking about the hormonal regulation, and we're talking about the allosteric regulation. So don't forget, we'll be talking about regulation of glycolysis using these three factors, or three factors of regulation. Number one, dietary; two, hormonal; and the last one is what? Allosteric. So how can we regulate it dietarily? First of all, don't forget, a high food with carbohydrates increases glucose, and when you have glucose is uptaken by the tissues, which is the muscle tissues or all the tissues, and what get induced with what? Glycolysis. Don't forget. So when it get induced with glycolysis, what happens? It will form what? ATP. So a high food, a high welfare state, or a high carbohydrate food promote what, or stimulate what? Glycolysis, and inhibit what? Glucagoneogenesis. So when you take food, you are storing the glucose in the liver as glycogen, and you are using some of them to form what? ATP. So it favors glycolysis. Don't forget. But when you don't take food, what will happen? It will inhibit glycolysis and favors what? Glucagoneogenesis. So a welfare state does what? Favors glycolysis. A welfare state favors what? Glycolysis, but inhibit what? Glucagoneogenesis. But fasting and strenuous exercise inhibit glycolysis and favors what? Glucagoneogenesis. Two is hormonal regulation. So anytime there's a presence of glucose in the blood, if it's a high glucose in the blood or a low glucose in the blood, what happens? Insulin is stimulated by the beta cells of Langerhans. So when there's a presence of glucose in the blood, when the glucose in the blood is high, or when there's even a little amount of glucose there, what happens? The beta cells secrete insulin to do what? To increase the uptake of glucose in the tissue for the purpose of what? Glycolysis. So when there's an increase in insulin to glucagon ratio, increase in insulin to glucagon ratio, what happens? Glycolysis will be stimulated. I get it now. Glycolysis will be what? Stimulated. Glucagon favors what? Glucagoneogenesis, and glucagon is secreted when we do not have glucose. When we do not have glucose in the blood. But here in glycolysis we have glucose, and we want to reduce the amount of glucose so that the glucose will be used to form what? ATP for our tissues. So we need insulin, not glucagon. So there will be increase in insulin production and a decrease in glucagon production, because an increase in insulin production favors glycolysis and inhibits gluconeogenesis, but a high glucagon secretion favors gluconeogenesis and inhibits what? Glycolysis. So this is the hormonal what? Regulation of what? Glycolysis. In the allosteric regulation of glycolysis, so we'll be focusing on the rate limiting enzyme, which is what? The step 1, step 3, and step 10. They are the irreversible reaction, and they are rate limiting, because of them glycolysis moves forward. So in our step 1, which is hexokinase glucokinase, insulin stimulates hexokinase to continue the reaction. Also ADP also stimulates the further uptake of more glucose for the purpose of what? For the phosphorylation to give what? Glucose 6-phosphate. So this is insulin and what? ADP are very, very important allosteric what? Activator of what? Hexokinase and glucokinase. So what inhibits these people? Glucagon inhibits hexokinase and glucokinase. Also ATP formation also inhibits hexokinase and glucokinase. They allosterically inhibit hexokinase and glucokinase. Also glucokinase is what? Inhibited by what? Fructose 6-phosphate. Why what? Hexokinase is inhibited by glucose 6-phosphate. Don't forget, they are inhibited by the same enzyme, which is ATP glucagon. But one problem here is that hexokinase is inhibited by glucose 6-phosphate. Why glucokinase is inhibited by fructose 6-phosphate. So both of them are stimulated, allosterically stimulated by the same thing, which is insulin and ADP, and they are also inhibited by the same thing, which is glucagon and ATP. But by the product of glucose 6-phosphate inhibit hexokinase, while the product of fructose 6-phosphate inhibit what? Inhibit glucokinase. The next rate limiting enzyme we'll be talking about is fructokinase, used in step 3. In step 3, fructokinase 1 transform what? Or phosphorylate fructose 6-phosphate at carbon 1 to give us what? Fructo-1,6 bisphosphate. This is important because ATP is converted to what? ADP here. In this reaction, it's still the same thing. They are allosterically activated by what? Insulin and ADP. But here there's also an addition. Because fructo-2,6 bisphosphate also also allosterically activate what? PFK1. Don't forget, I said it in the reaction whereby fructose 2,6 bisphosphate also stimulates the enzyme PFK1 to continue the production of what? Fructose 1,6 what? Bisphosphate. So we have three activators. The first activator is what? Insulin. Second activator is what? ADP. Third activator is fructose 2,6 what? Bisphosphate. So what is inhibiting here? There's now the same enzymes are inhibiting, which is what? ATP, glucagon. But here there's one important inhibitor here, which is what? Citrate. Citrate is formed when what? Acetyl CoA is combined with what? Oxaloacetate. This citrate here inhibits what? PFK1, which is phosphofructokinase. Again, citrate inhibits what? Phosphofructokinase, because citrate is gotten in TCA cycle when acetyl CoA combined with what? Oxaloacetate. Don't forget. So we have three stimulator and we have three inhibitor. The addition inhibitor here is what? Citric acid, which is formed in TCA, and the addition activator is the what? Fructose 2,6 what? Bisphosphate. In the step 10 of glycolysis, which is what? Pyruvate kinase. Pyruvate kinase is another important rate limiting enzyme, because when you increase this enzyme, when you increase the formation of this enzyme, what happens? PEP, phosphoenolpyruvate, will form what? Pyruvate. So who stimulates this reaction? The same people, which is what? Insulin, ADP, and also fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate, insulin and ADP stimulates pyruvate kinase. But which enzymes, what products also inhibits this? Don't forget, the same people, which is what? ATP, glucagon. Also we have long chain fatty acid. Long chain fatty acids also inhibit pyruvate kinase. Also we also have citrate. Citrate can also inhibit what? Pyruvate kinase. So these are the important enzymes that is inhibiting what? Pyruvate kinase. So we have said it for the step 1, step 3 and step 10.